The bio unit basically just introduces us to all of the body's physiological processes and how those things affect our behavior. It's 8 to 10% of the AP exam, and some of the main areas or places of interest that we talk about include the brain, the nervous system, neurons, and neurotransmitters. And some students find this unit really hard. If you haven't taken a bio class before, you might not know a lot of the terminology for the brain parts and for the neurotransmitters. But if you have taken bio before, some of you might think this is an easier unit. So it really just kind of depends where you are in your studies. My students don't take AP Psych typically until junior year. I don't know, how old are you guys? What grade are you guys all in? Let me know in the chat. I just like to see where everyone is coming from and where we're all at. Junior, yeah, I think that's fairly typical. But yeah, let me know. So, ooh, my computer is being fussy. Here we go. All right. So this first thing, this is a diagram from the Myers book. And we're going to spend some time talking about just communication within the neuron. And so there's kind of two pieces here to understanding how neurons work. You have to understand how it works in one piece, like how a neuron communicates inside of itself. And then later, we'll also talk about how a neuron talks to other neurons or other body parts. And so both, I think, are important. The first thing is you just have to know the pieces of the neuron. Occasionally, there might even be a diagram on the AP exam that says, what is this? And it might point to something out here or here or whatever and ask you to label it. And so I'm sure your teachers maybe do. My students, we build candy neurons. So I give them pretzels and gum and Twizzlers and dots and all kinds of different types of candy, and they have to build a neuron out of that candy and kind of figure out how the different parts could be represented by the different pieces of candy that they were given. Like a marshmallow might represent the cell body. The Twizzler might represent the axon, those sorts of things. So however it works for you to memorize the parts of the neuron, you do need to know each part and kind of what it does. And so how a neuron talks to itself or takes information is it starts with the dendrites. Those are those little spiny things towards the left here. And they basically like suck in or receive messages from other cells. So that's where the information enters the cell. It takes it in through the dendrites, and then it takes it into the cell body, and that's kind of the life support center of the cell. You don't need to know a ton about the cell body except that it's where the dendrites pass the information through. And then probably the most important part that you need to know about the neuron is the axon. And so the message continues down the axon there. That's the like ropey thing here in the middle. And this is done in like brief electrical impulses. It's called action potential. We'll talk about action potential specifically on another slide. But basically it's like a little flicker and it's like bzz, the message goes bzz, 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 and the message travels down the axon. And there is a coating on some, not all, axons called the myelin sheath. And basically that's just like a conductor. It makes it go faster. So it makes it from going like bzz, bzz, to going like bzz, 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 bzz. It helps speed up the communication. It goes a little bit faster. It speeds up the neural impulses. And then basically the end goal here is for the information to reach the terminal branches. It sometimes is also called the axon terminals. And so those little buttons at the end there of the terminal branches are where the next message shoots out from. And then it would just continue how we started with this one. So basically, it only travels in one direction. That's important too. It only goes like dendrites that way. It can't go backwards. But the real goal here, the number one goal of why a neuron exists, is to communicate with other cells. That's why it exists. It wants to talk to other cells and pass its message along. Typically, those other cells are neurons, but sometimes not. We'll talk a little bit about that. So its goal is to communicate. So if you understand kind of how that message gets into the dendrites and down the axon, you are in a good place so far. So let me know if you have any questions about that. We're going to look and zoom in on the axon now and talk a little bit more about action potential specifically and how that message works. So we're zooming in. You see the neuron picture there. This is again from the Myers book. And so this is maybe the trickiest part of, oops, there we go. Maybe the trickiest part in how a neuron goes. So an axon doesn't just fire willy-nilly. It has to have a certain amount of stimulation. It will not fire until it reaches whatever that level is. The level is not super important for you to understand right now, but you just need to know that it has to be there. It has to reach a level. So while the neuron is waiting to be stimulated, it's polarized. And so you can kind of see those pluses and minuses that are in the first part of the diagram there. And so outside of the neuron, there's a high level of sodium positive ions. And then the resting fluid interior contains mostly negative ions. So it's positive on the outside, mostly negative on the inside. And so it's just hanging out, right, ready to just fire. And we know that when something like depolarizes, what happens, it wants to like reassess to its neutral spot. So what happens is when that specific level of stimulation is reached, the dendrites are pulling in those messages. Once enough of that signal is reached, the cell starts to depolarize. And so what ends up happening is sodium ions on the outside push in, and then it pushes out potassium ions, and the cell is depolarizing, then becoming positive inside, negative outside. So that is what the diagram here is showing you. And basically, the cell wants to go back to its neutral state. So as the positive is rushing in, the negative is rushing out, the positive is rushing in. It just kind of goes like this, in, out, in, out, in, out, in, out. That's what pushes the message down the axon. To me, this is one of the most complicated things that we will talk about. So if something is not making sense, please tell me and I will pause and think about a different way to say it, or we can look at a different diagram. We can figure things out. But I really want to make sure you understand this. So basically, the depolarization is what's pushing the message along the axon. And then it's trying to repolarize itself. Positive in, negative out, positive in, negative out. And that is how the message travels down the axon. So once the axon returns to its polarized normal state, the message is done and it's reached its sending point. And so it always goes, from the side with the dendrites to the end of the axon terminals. And then we will talk a little bit more about this kind of needing a certain amount of stimulation to fire, because that's kind of important too. But we'll get to that in a little. So that is the basic gist of how a neuron talks within itself. But like we said, the main goal of a neuron is basically to communicate with other neurons. And so how does it do that? We've seen how it goes within itself. How are we going to see it continue to talk then to the next neuron and the next one? So this is another diagram from the Myers book. But what we are seeing here is kind of a close-up of where the talking takes place between neurons. So this little, can you see my mouse? I don't think you can. When I do this, can you see my mouse at all? No, yes, no, I don't know if you can see my mouse. So basically those little buttons at the end of the terminals that we saw on the last slide, the little kind of balls, those have neurotransmitters in them. And so we call those synaptic vesicles or maybe terminal buttons. Sometimes it depends what your book is calling them. But basically that message goes down the axon and it reaches those axon terminals, those terminal branches, and then little, I call them balls. They're not really balls, but little balls, little neurotransmitters are released. And what happens is those neurotransmitters cross a gap to the next neuron. And so they try to attach to the dendrites of the next neuron. So that's, remember what we talked about, how the message gets sucked in. And this is important. Neurons do not touch. they're super duper close together on like a microscopic level, but they do not touch. There is like a little gap where these little balls or these neurotransmitters need to cross to reach the next neuron. So they cross the synaptic gap, these little balls, and then they bind to the receptor sites of the next neuron. And they try to, sorry, my dog's in the background. You can see his butt there. Oh, there's his head. Hi, Moose. And they get sucked into the next neuron. Not all of those always get taken in though. And so what happens is if this is the sending neuron and it's shooting out its little balls and then this neuron's catching them, if there's any left over, they get sucked back up into the sending neuron. So anything that's left over gets taken back by the original neuron, and this is called reuptake. Drugs can interfere with this process. Many antidepressants, anti-anxiety meds, those are basically all focusing on that premise of reuptake. They prevent it from happening. We'll talk about that a little bit later. So yeah, basically a neuron shoots out little balls, vesicles, neurotransmitters, and then the next neuron sucks it in and takes it and then passes the message along, and that's how they continue to communicate. And we're talking about this very slowly, but think about how quickly you can just like touch your head or think about anything. Neurons are firing at like the blink of an eye so fast. We don't even really have time to think about it and consider it, but to understand it, we have to break it down into teeny tiny little steps so that we know all of the pieces that go into it. And it's actually, I think, pretty amazing if you think about all of this is happening inside your bodies every second just to like keep you alive and keep you functioning. It's a pretty cool process, I think, to think about. So how does a neuron then decide if it's actually going to fire? And so I like this analogy of a flushing toilet here. I think it's useful for you to kind of think about this when you think about a neuron firing. So first and foremost, a threshold has to be reached. We talked about that. A neuron will not fire until a certain amount of threshold is reached. And if you think about that, think about a toilet handle. If you just push it a little bit, it doesn't flush, right? You have to push it a certain amount for a toilet to flush. So a neuron and a toilet are very similar in that regard. You have to give it enough pressure or reach a certain threshold for it to fire or to flush. The next important comparison between a neuron and a neurotransmitter is that you can't just partially flush a toilet, and a neuron can't just partially fire. It either fires or it doesn't. A toilet either flushes or it doesn't, but you can't stop it, right? If you reach the threshold with the toilet, pushing the plunger, you can't all of a sudden stop it from flushing. Once it starts, it finishes the process. Once a neuron starts to fire, it finishes the process. Kind of similar. The gross or negative, as in the negatively charged particles, the contents are pushed through. So the negative ions are pushed through when a neuron is firing, just like the gross or negative contents are flushed through a toilet. Also, luckily, a toilet and a neuron can only flush in one direction, right? It would be pretty gross if a toilet could go the opposite direction. You'd end up with gross things all over you. And so just like a toilet, a neuron only goes one way. It only flushes in one direction. And then lastly, a toilet and a neuron are similar because a toilet cannot flush again until it has had time to reset itself. And a neuron can't fire right away. A neuron has to, we talked about, kind of reach its state of repolarization before it's ready to fire again. And so think about it, that's like the time period when the toilet is like running and it's filling back up. Your neuron takes a second to also be ready to go again. Again, in a neuron, it's like it feels instantaneous, but there is a brief pause before it can fire again. Hopefully that analogy is helpful to you. It seems to help my students when thinking about all of the things you need to know about a neuron. And it seems so silly. We're spending so much time talking about this, just teeny tiny little things, but they're so important to understanding how the brain and the body function. So let's take a just 5-second break because I feel like this stuff is heavy. If you have any questions, please feel free to put them in the chat or in the Ask a Question tab. And then for everyone that's in here right now, can you just give me a shout out to where you're at, what state, what country, what city? Don't be shy. This goes better when we all are here and participating. So where is everyone coming from tonight? I live in Madison, Wisconsin. And I teach in a suburb of Madison called Middleton. It's kind of nice here tonight. It's like 70 degrees and sunny. Texas. Ooh, I bet it's hot in Texas. Anyone else? Where are you guys at tonight? See if my dog can make an appearance. Do you want to say hi? Come here. This is Moose. He's going to say hi to everyone. Okay, he's giving me kisses. South Carolina, Iowa. Ooh, Iowa. Are you an Iowa or Iowa State fan? We're going to have a big game coming up between the Badgers and the Hawkeyes. All right. No questions. You guys totally get neurons and neurotransmitters. I have some multiple choice questions that we'll go over at the end, but I'm going to move on now to talk a little bit about specific neurotransmitters. Ooh, Hawkeyes. Ooh. We'd be rivals, I think. I'm a big Badger fan. So we're going to talk about neurotransmitters, and then we'll kind of come back around and do a few review questions at the end to see where we're at. All right, so when we're talking about neurons firing and talking to each other, there's kind of two ways that they do this. One is through an electrical synapses, and one is through chemical synapses. And so basically, an electrical synapse is used for things that have to be kind of constant. So like I need to breathe and have a heart to be alive, and I need those things to happen quickly and consistently. So those are like quick, efficient, and constant things. Our electrical synapses are just there, always happening. On the other hand, we have chemical responses in our bodies. This is more having to do with neurotransmitters. This is what we're more concerned about right now, is the neurotransmitter side. And so this is where I was talking about the little balls are fired across from one neuron to the next. Those are chemical. These are more abundant, but they're also more precise. Like me reaching and touching my nose sends a different message than like me waving my hand at you. So it's much more precise and refined for whatever it is that I'm doing. It's very selective. My brain and body knows how to like message me and tell me to do those things, versus the electrical, which is just like bzz, 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 constantly breathing, heart beating, keeping me alive, that sort of thing. So we're kind of more concerned with the chemical ones tonight. So some specific, excuse me, some specific types of neurons then that exist are sensory / afferent. That means the same thing. You just have to know both names. Or motor and efferent. And so the sensory neurons are the things that if I touch a hot stove, it inputs that message to my brain and says, hot, take your hand off the stove. So it carries the messages in, it's input. And how I tell my kids to remember that is that A for afferent is the first letter of the alphabet. So a message has to come in first before you can respond to it. A is for afferent. A is the first letter. You have to bring a message in before you can take a message out. And so it carries all of my sensory neurons. I would have like my fingertips have a lot. My lips have a lot of sensory. My tongue, because it's all taking in refined. It has to be like refined messages. So those messages go inward to my central nervous system, which then sends it to my brain for processing. My motor or efferent neurons take the messages from my brain and central nervous system out to like my hands and my feet and my legs and my head and whatever I'm doing. It's an output message. It's telling my body what to do. How I tell my kids to remember this is efferent sounds like effort, and it takes effort to move or respond to something. And so that's what motor neurons do. Again, we have a lot of motor neurons in our fingertips, places that we use a lot, our lips. That's probably two of the biggest places, fingers, mouth, lips. But yeah, so just make sure you know those two different types. And I think the A and the E thing is useful for remembering that. Okay. So now the specific neurotransmitters, these are the little balls that pass between neurons. These are chemical in nature, and they transmit. Oh. Nina, does same mean like S for sensory, A for afferent, M for motor, E for efferent? That makes sense. That's another good tool for remembering it. Any sort of mnemonic device that you can use to remember would be really useful. Afferent. Okay. Yeah, any sort of mnemonic device that you can use to kind of help you, I think is really useful. So if that works for you, also, yeah, same for, yeah. Cool. Good. So neurotransmitters specifically are chemical messengers that transmit signals from one neuron to another. We've talked about them a little bit already, but now we're going to get into the nitty-gritty details. If any of you guys ever have other little mnemonics, like what Nina posted or what your teacher tells you different, please post them in the chat because those are so useful and it might just like make things click for other people. So for neurotransmitters, there's like 100 different types. Don't worry, for AP Psych, you only need to know like 6 or 7. There's a lot. And each one has like a unique little shape and size, which basically means it matches or fits with like a unique shape or size on another neuron. And so, like I said, they're selective and they're specific, and there's different kinds for different things. And so, depending on which neurotransmitter is firing or how many of them are released from the sending neuron to the receiving neuron, depends then if the neuron reaches action potential. So these neurons are what decides if that threshold is reached for like the flushing of the toilet, for the firing of the neuron. However many of these reach and are received, bind to the receptor sites and then make the next neuron fire. Okay? And the big ones that we need to know, I guess first you need to know the difference between neurotransmitters and agonists and antagonists. So neurotransmitters, like we've been talking about, they come from within the body. They're naturally occurring things within the body. They can excite or inhibit our neural communication, meaning it can tell my body to fire or it can tell my body to slow down. And then the examples listed, acetylcholine, dopamine, we'll talk about those on the next few slides. But these are all things from within our bodies. But I mentioned earlier, you can also take like prescription medications or illicit drugs that mimic some of these responses. And those are called agonists and antagonists. And so agonists or antagonists are drugs and other chemicals that come from outside our bodies, but yet mimic what a neurotransmitter does or block a neurotransmitter from working. So an agonist binds to receptor sites and mimics its effects. So basically, maybe you don't get enough serotonin. And so maybe you've heard of people taking SSRIs. SSRIs are selective serotonin reuptake inhibitors, which basically those are like antidepressant medications. And what they do is they can help you like experience more of it. It can prevent the reuptake, so it prevents the serotonin from getting sucked back up into the old neuron, or not the old, the sending neuron, and it makes its effects last longer. Antagonists bind to receptor sites and actually block it. You're getting too much of a neurotransmitter and it's causing problems in your body. You can take a medication to kind of block it or slow it down. For that, think of like, in English class, if you're reading a story and the antagonist of the story is the person who tries to like prevent the hero from being successful. Well, the antagonists in your body are preventing or blocking the effects of a specific neurotransmitter. So heroin acts as an agonist. It like binds to receptor sites and it like mimics, what does heroin mimic? I want to say it's dopamine. Heroin. Not positive, but heroin mimics one of the neurotransmitters in our body, which gives you this like up feeling, and you feel so good because you're getting like a rush of this like feel good neurotransmitter. Does that make sense? So then the basic 6 neurotransmitters that I just had listed, acetylcholine, ACH is how you might see it abbreviated. And like a lot of these neurotransmitters do a lot of different things, but for like AP Psych, the few sorts of like tasks that you have to know are basically listed here. Because if you like Google acetylcholine, there's going to be like, whoa, like pages and pages of information. The gist of what you need to know are just these few bullet points. So just make sure you understand these. So acetylcholine has to do with muscle action, and it enables our learning and our memory. So sometimes we think that acetylcholine might be linked to Alzheimer's or other illnesses that have to do with like deterioration of some of those functions. And so how I remember acetylcholine is macho man, because the ACH in macho, you know, like muscle action and movement, things like that. That's how I remember that. ACH has to do with muscles and movement and voluntary action, things like that. Dopamine has to do with voluntary movement specifically, and then reward centers in our brain for pleasure. And so, yeah, heroin, I believe, is a dopamine agonist. What that means is if you do heroin or a drug like heroin, it actually like mimics the dopamine in your brain. And so the dopamine makes you feel good. When you take heroin, it's like feel good times like 1,000. Your brain and your cells are going crazy because you're getting this rush of extra dopamine. It's mimicking what dopamine does. You might think, well, why is that problematic? Like, you want to feel good, right? The reward centers give you pleasure and a good feeling. But what happens is when you take a drug like heroin, your body says, whoa, I'm getting so much dopamine. It stops making dopamine yourself. So your body stops making the natural dopamine because the heroin is replacing that. So then what happens when you're like, okay, I need to like quit taking drugs, it's not healthy. What happens is you quit the drugs, but now your body isn't producing anything to make you feel good either. And so you get those nasty withdrawal symptoms. Your body just crashes because it's given up making the dopamine, the feel good, because you were replacing it with fake kind of feel good. And then what happens is, and why people get withdrawals, is because their body's craving that feeling and it's not producing it on its own, which is why a lot of people relapse, because they can't feel good again without it. And we have other medications and stuff that can kind of help with that, but that's kind of the gist of how like addiction with some of those drugs works. Serotonin is another important neurotransmitter. It has to do with sleep, eating, and mood. And so the the 2 O's in serotonin go with the 2 O's in mood. That's how I remember it. And serotonin is specifically related to depression. People who are diagnosed with depression, like I said, are often lacking or not getting enough serotonin. And so if you have ever heard again of SSRIs, selective serotonin reuptake inhibitors, they basically prevent the serotonin from getting taken back into the neuron. They make it stick around longer so that it is absorbed by your body. That's how antidepressants work. Norepinephrine controls alertness, arousal, and mood. So here we've got 2 again that kind of focus on mood. So you can kind of see how they work together. It's not just one versus the other. It's also an agonist, and it creates a euphoria. So when you get that like adrenaline pumping runner's high, anything like that, that kind of comes from the norepinephrine. It's kind of this euphoric, make you feel good sort of neurotransmitter. GABA is an inhibitory neuron, and if you know what inhibit means, it means to like slow down or stop. And so basically it tries to control and prevent too much of everything going on. It wants to like keep you regulated, keep you in like a calm and regular place. It doesn't want your brain to be like overstimulated and firing back and forth and going all out of control. And then glutamate is an excitatory neuron, It's involved in memory, and the biggest thing with glutamate is it might be linked to migraines and seizures. So if you think about it, it's an excitatory neuron. It makes things happen. It's go, go, go. But if you have too much go, go, go happening in your brain, it might make sense why you'd have a migraine or even a seizure, because there's so much firing. If it's like out of control or if it's not functioning properly, it could cause a migraine or a seizure or things like that. So those are the 6 main neurotransmitters that you need to know for AP Psych. The other thing is that they come up again and again. It's not like, oh, you need to know these just this one time and then we're never going to talk about neurons again, or neurotransmitters again. They will come up over and over again. And so the nice thing is that even if you don't feel super confident about them right now, they will be reviewed when we talk in other units. When we have abnormal psych, we'll talk about them. When we have just a bunch of different things, they come up. So I'm going to talk a little bit then about multiple choice questions and strategy, and then I do have 5 review questions that we can just kind of go over just to see if you feel confident in what we talked about tonight. So, and if you have any extra strategies, put them in the chat, because I'm always looking for new things to tell my students. So first and foremost, I always say never skip questions, especially on the AP test. It's 100 multiple choice questions. And if you accidentally skip one and then you like forget to not bubble, imagine how all your bubbles could be off. So I always tell my students, even if you're not sure, always just bubble something and then make like a little mark next to it to remember to come back to it later. But I would never skip a question, because you just risk getting your whole bubbling off and then your whole test is messed up, and that would suck. There's also no penalty for guessing in AP Psych. So like, why not at least try and guess an answer? It doesn't hurt, and you have like a 20% chance of getting it right, so you might as well. When you're reading the question, try to highlight the main idea or what the vocab is or what the question's getting at. You won't have time to do this for every question. Some of them are pretty straightforward, but on some of the more wordy questions that are kind of long-winded, it might help to kind of get to the root of what they're asking. And then lastly, this is a timed test. So if you are running out of time, I say always bubble the same letter. That came from some kids who do ACT prep with some prep companies. They were told that. And so I'm guessing it's because it has to do with, like, if you're skipping around switching letters, you could accidentally miss you have more of a chance of getting them wrong. If you bubble the same letter, you are bound to at least get a few of them right. So try that. If your teachers give you any other strategies, let me know in the chat. So in the poll, you'll see there's a poll response question here at the bottom of your screen. And so I would like you to put what you think the answer is to question 1. It's anonymous, so you don't have to be shy about putting a answer in, but we're going to just go over some questions just to kind of review and see if you remember or understood everything we talked about tonight. So question 1 says, in transmitting sensory information to the brain, an electrical signal travels from the blank of a single neuron. So within a single neuron, how is this message traveling? From what to what to what? Put your answer in the poll. Give you like 30 seconds and then we'll go on. It's anonymous. Don't be afraid to get it wrong. So yeah, it looks like you guys have got this. The answer is D. Remember, it starts with the dendrites. That's what sucks in the message into the cell body and down the axon. Good. So you can use the same poll now, and you can just change your answer for question 2. This one we did not talk about specifically, but it's a good question to kind of understand the purpose of I just gave you the answer. The slowdown of neural communication in multiple sclerosis involves degeneration of what's the answer? I started to say it. Oopsies. So slowing down in the neuron. What might cause it to slow down? So yeah, the answer here is myelin sheath. The myelin sheath is that coating that wraps around the axon, and it's what speeds it up. It's kind of like an insulator cable to make it fire faster. And so it's suspected that in multiple sclerosis, that myelin starts to like degenerate and go away. And so it makes people with multiple sclerosis have a harder time like having that speed and that response time. All right, question 3. Within a single neuron, the action potential, which of these is true? Within a single neuron. So the answer to this one is A, travels in one direction toward the axon terminals. D, it's kind of tricky because it says within a single neuron, the action potential. D is involving more than one neuron. If it's crossing the synapse, then it's talking about multiple neurons. So it's kind of a tricky question. A is where it's within one specific neuron. Okay, 2 more questions. Psychoactive drugs interfere with normal neural transmission. Where does the interference take place? This is a tricky one. So if you're doing heroin or another psychoactive drug, how is it impacting your neurons? Where is it impacting your neurons? This is probably the hardest one. You guys are doing good. Yeah, the synapse. So it's the little gap between the 2 neurons. It's impacting how the one neuron is firing it and what's happening to those neurotransmitters. Are they finding receptor sites or are they being blocked by the psychoactive drugs? So it's happening with the synapse, the gaps between neurons. Good. And then question 5, a person with schizophrenia may have an overactive dopamine system. Drugs used to treat this disorder prevent the action of dopamine by keeping it from binding to its receptors. These drugs are good. Antagonists. Yeah, remember they block, they prevent, just like the antagonists in a story.